What is in the Water by Bailey Schatza for the purpose of Chemistry 120N, Lab Section 2. Water is everywhere around us, and it is primitive for human survival. So, it is important that the water we humans are consuming and that are around us in our surroundings is clean. So, the purpose of this lab was to determine unknown compounds that were in our drinking water samples. So the sample explored in this experiment is sample C, which is also known as vial 31. The technology used were NMR and IR spectroscopy. The NMR spectroscopy showed the number of peaks and the ratio of areas, which was used to help determine which compounds. And furthermore, the IR spectroscopy showed the possible functional groups, which also helped determine which compounds were in the water. So, as you can see here, this is an NMR spectrometer, and then here is an IR spectrometer. So, the experimental technology used, as I just explained, the full formal terminology is the NMR spectrum was the Anzali Instrument Inc. EFT60 NMR spectrometer with 1.4 Tesla permanent magnet operating at 60 megahertz proton frequency. It had PNMR spectrometer control and data acquisition software with one proton scan, NUTS Pro software, which also used to display the spectra. And then the IR was the Thermo Scientific Nicolette IS5 FT IR spectrometer with ID7 ATR with a volatile sample cap. A diamond crystal ATR sample accessory with a metal cap was also utilized. The OM NIC instrument operating software displayed the spectra, producing 16 sample scans. So, the procedure. The IR spectra was taken and ran, and then the functional groups were presented to us through graphs. And then the NMR was ran, and the peaks and ratio of areas were displayed. And then through the two, tables and figures were created, and then a comparison of the results was made, therefore leading to the identification of the unknown sample. This is a table depicting the IR possible compounds. So, for example, for a methanol compound to be present, an OH group and a CH group would have to be present in the IR spectra. So this is just a table depicting all of the possible ones that was used later whenever my graph was made to be able to depict the compound. So this was my graph that was produced, and so for vial 31, as you can see, there is a clear OH group in the range between 3,500 centimeters to the negative first and 3,100. And then there's also a CH single bonded carbon group present in the 3010 to 2800 range. This is a table of the NMR possible compounds. So again, for example, methanol, the graph would show 2 peaks, and then the ratio of areas would be 3:1. This was my spectra that was produced. As you can see, there is a 6:1:1 ratio, and there were 3 peaks. So the information between the OH and the CH, along with the 3 peaks with a 6:1:1 ratio, were then compared, as stated here. So Table 1 and Figure 1 were compared for the IR, and then Table 2 and Figure 2 were compared for the NMR, and the IR predicted that methanol, ethanol, 2-butanol, and 2-propanol were all possibilities. For the NMR to clearly indicate which one or further indicate which one may be present, the only possible compound with 3 peaks and a 6:1:1 ratio would be 2-propanol. And since that was also a possibility in the IR, by the comparison of the two, the compound was able to be identified. So again, the identification for unknown sample C, vial 31, was 2-propanol. And then just some health risks for 2-propanol. If you consume it in your drinking water, it might lead to the slowing of heartbeat, nausea, stomach pain, confusion, dizziness, low blood pressure, respiratory irritation, central nervous system depression, dilation of the blood vessels, vomiting, and disturbed motor responses. So, as can tell, those are all something that you don't want for health risks, and therefore you do not want to be consuming 2-propanol. And then just some environmental effects. It is harmful for the environment due to its high flammability and its mobility due to its volatility, meaning that it can move pretty quickly and pretty intensively throughout water, so it can spread. And then in order to use 2-propanol, explosion-proof electrical ventilating light and lighting equipment is advised and recommended. And then, just in conclusion, the quality of drinking water is important, especially when considering human health. So, the IR and NMR spectroscopy are extremely useful technology that allow people to understand the quality of their drinking water and to make sure the quality of it is also safe for human consumption. And then, again, just the IR and NMR were able to identify the compounds. And 2-propanol is hazardous in both human consumption and drinking water. Thank you. And these are my references.